So cell respiration is related to photosynthesis in a lot of ways. If we look at photosynthesis, if we go back to photosynthesis for a second, in photosynthesis, carbon dioxide and water got combined and built glucose. So photosynthesis was the process of capturing the sun's energy to make glucose. But glucose, and I've given this example before, but imagine if you guys all needed to run little cell reactions, and each cell reaction that each person needed to run needed a quarter. Glucose would be sort of like me having a $20 bill. So by taking the glucose, my $20 bill, and getting change, or a bunch of quarters, I could give each person a quarter, and they would have the amount of energy that they would need to run their personal cell reaction. And that's basically what's happening in cell respiration. Glucose is getting broken down, and the energy is being extracted from it. So in a lot of ways, it is kind of like the opposite of photosynthesis, but it's a completely different process. It's not photosynthesis running backwards or anything like that. It is its own process. So it extracts the energy from glucose, breaks it into ATP, or makes ATP, breaks glucose into carbon dioxide and water. Technically, cell respiration can happen with or without oxygen, but when I just generally say cell respiration, I'm referring to the full process in the mitochondria with oxygen, even though there is such a thing as anaerobic respiration, which we'll talk about in a second. Also, you don't have to use carbohydrates for cell respiration. If you want to lose weight and you go on a diet, your goal is to burn fat. You can use fats for cell respiration. You can also use proteins. If you go on a starvation diet, you'll start burning up your muscle to make ATP. So fat, carbohydrates, and proteins can all be used for cell respiration, but since it's generally run with glucose, that's what's floating around in your blood, we usually see this formula with glucose. A quick reminder, and this is important for testing purposes, that when glucose does break down, the carbon and the oxygen from glucose become the carbon and the oxygen in CO2. This was important in photosynthesis also. I told you that there could be questions about where does this hydrogen come from? Where does this oxygen go? The oxygen, the free-floating oxygen that we see coming in here, this oxygen ends up in water. And the hydrogen in glucose here also ends up in water. So it is important to note, although it's not wasn't already color-coded here, it was color-coded in photosynthesis, and the color codes match. In photosynthesis, it was exactly the same thing. That water was split, and the hydrogen from water ended up in glucose. The oxygen from the water ended up being released in photosynthesis. So, and the carbon and the oxygen and carbon dioxide ended up as part of glucose in photosynthesis. They go back to being carbon dioxide. So it's theoretically possible that if you had a mouse and a plant in a jar, that the mouse could be running cell respiration and release carbon dioxide, and that plant could then take that exact carbon atom and it could become part of the glucose in that plant. It's very possible. And then if the mouse was to eat the plant, the mouse could end up eating that carbon atom again, and you'd see the whole cycle of the carbon dioxide going into the sugar and back again between the mouse and the plant. All right, so and the purpose of this again is to extract energy, to extract and make ATP. But this is the overall formula for it, even though it's a multi-step process. So, any questions so far? Very general so far. Okay. So I'm going to clear this. So the breakdown of glucose does not happen in one step. It happens in a whole bunch of steps. Just like how photosynthesis was a bunch of steps, cell respiration is also a bunch of steps. One of the really important things that's involved in the process of cell respiration are coenzymes. They are NAD and FAD in cell respiration. And NAD and FAD, when they pick up hydrogens, become NADH and FADH2. Do you remember what the coenzyme was in photosynthesis that picked up hydrogen? It was very similar. Exactly. It was in photosynthesis, it was NADP. and it became NADPH, and then the H got transferred to glucose. So one way to remember, they're all coenzymes, but one way to remember the one in photosynthesis is think of the P for photosynthesis. NADP is the one that's in photosynthesis. NAD and FAD are in cell respiration. But they are coenzymes, and their whole job really is to pick up hydrogens and then later on release those hydrogens. When they pick up hydrogens, that's temporarily storing energy that's being reduced. When they get rid of the hydrogens, they go back to being oxidized. And just like in photosynthesis, what will happen here is NADPH will later give that hydrogen to glucose in the Calvin cycle, and then the NADP would get recycled back, and then it could go through the light reactions again. So it's bringing the dropping off the hydrogens, going back again, dropping off the hydrogens, going back again. The same thing is going to be true for these. They pick up hydrogens temporarily. They are going to take them to the mitochondria, drop off those hydrogens, and get recycled, and they can be used over and over and over again. Just like an enzyme can be used over and over again, these coenzymes can be used over and over again. All right. So NAD, when it picks up the hydrogen, it temporarily holds energy. When it later goes back and gets rid of the hydrogen, it will release the energy. This number is not important right now, but just to give an example, when NADH goes back to being NAD, going back to the oxidized form, it actually will generate 3 ATP. And FADH2, when it goes back to its oxidized form, actually generates 2 ATP. So each one of these coenzymes, when they get rid of those hydrogens, can generate multiple ATPs. And so this is going to be one of the major things that happens in cell respiration. Okay. I believe on your notes this is a table. There are 2 ways that this can work. So, like I said, even though in general, when I use the term cell respiration, it refers to a general process of breaking down sugar to make ATP involving oxygen, technically there are 2 ways you can get energy out of sugar. You can do it anaerobically, meaning without oxygen. So anaerobic, it means without oxygen. Or you can do it aerobically. So we believe anaerobic pathways evolved first, which makes sense because scientists speculate that early Earth didn't have any oxygen. So there couldn't have been aerobic respiration. There could not have been organisms that need oxygen if there was no oxygen on the planet. So most bacteria, a lot of the ancient bacteria and the older bacteria, run anaerobic respiration without oxygen. Both anaerobic without oxygen and aerobic, both of them can begin the first steps of that type of cell respiration. They both start in the cytoplasm. However, aerobic respiration, the one with oxygen, finishes in the mitochondria. It turns out, and this will come up later, but anaerobic respiration per glucose, I'll just add this, even though, like I said, it's going to come up later. You only make about 2 ATP per glucose in anaerobic respiration. Whereas, if you run aerobic respiration, per glucose, you can extract 36 to 38 ATP. So the reason why you'll have brain damage after 6 minutes or so without oxygen, you can still do cell respiration. The problem is you can only do the anaerobic portions of it. And you don't make enough ATP per glucose. It's basically like most of the energy in glucose ends up in the products and you're not able to extract it. So you won't make enough energy to keep your cells alive. The other problem with anaerobic respiration is that the byproducts of it are typically toxic to us. Bacteria, when they do anaerobic respiration, make things like alcohol. That's how we basically make beer and wine and stuff like that, is bacteria running anaerobic respiration. That's their byproduct. We make lactic acid. So when you're working out and you're not getting enough oxygen, your muscles start burning, it's because you're running anaerobic respiration only, and you build up lactic acid in your muscles, which will then eventually cause them to collapse. And not only that, but if you don't flush that out, it'll leave soreness and irritation. So lactic acid's not a good thing. So anaerobic makes less ATP than aerobic. But bottom line, they both start in the cytoplasm with a step called glycolysis. Anaerobic only happens in the cytoplasm. Aerobic finishes, the last steps of it will happen in the mitochondria. So usually when we talk about cell respiration and we say, oh, what organelle is involved in cell respiration, we say the mitochondria. Can you do cell respiration without mitochondria? Technically, yes. But you can only do the anaerobic portions of it, which is not enough to keep you alive. So any question on the comparison between anaerobic and aerobic? All right. Get rid of this. All right, so not only is there anaerobic and aerobic respiration, but there are two ways to make ATP. The first way, remember the word phosphorylation just means to add a phosphate. So don't let that word phosphorylation confuse you. But remember, what's really happening in making ATP is you're just sticking that third phosphate on to ADP. So ADP, adding a phosphate makes ATP. That's what's really happening in cell respiration. So substrate level phosphorylation, the name comes from the fact that the way that the phosphate is put on ATP is that literally an enzyme takes a phosphate off of a substrate and sticks it on ADP. And I'll show you a picture of it on the next page. This method of making ATP is not very efficient. It doesn't make very many ATP. In fact, it doesn't require oxygen either. So typically anaerobic respiration, if you're running anaerobic respiration, you are only making your ATP by this method. Some bacteria only use substrate level phosphorylation. They make all their ATP this way. How can they get away with that? Because they're bacteria. They're a single tiny cell. So even if they're only making a couple of ATP per glucose, it's probably enough to keep them alive. It's not enough to keep a eukaryotic cell alive, and it's definitely not enough to keep a multicellular organism alive. So this is called substrate level phosphorylation, and again, all that's happening is a phosphate is being taken from a substrate, a thing. If you remember enzymes we talked about before Christmas, a substrate's just the thing that the enzyme reacts with. The phosphate's literally taken off the substrate and stuck on ATP. And again, this type of phosphorylation does not require oxygen. So if there's no oxygen present, you would only be able to make ATP by this method. And you can't stay alive very long making ATP only by this method. We rely partially on this because glycolysis makes ATP this way, but the remainder of the processes in cell respiration make ATP in a different way, which is a much more efficient way of making ATP. All right, so any questions on substrate level phosphorylation? Okay, I have a picture of it as well on the next slide. So this is literally what it looks like. Substrate level phosphorylation. This green thing is the substrate. It has a phosphate on it, and the phosphate gets transferred to ADP, making ATP. That's literally what happens in substrate level phosphorylation. It's pretty straightforward. The other kind of phosphorylation is the one that's sort of complicated. It's called chemiosmotic or oxidative phosphorylation. This method, which is much more efficient, relies on redox reactions. And this method happens in the mitochondria of us, technically the inner membrane of the mitochondria, but there are some bacteria that can use their cell membrane to run this process, which makes sense because where did we say mitochondria originally came from? They were bacteria that lived on their own, remember endosymbiotic theory, that got engulfed. And there are still cells today that behave like a mitochondria. So their outer membrane behaves kind of like a mitochondrion. So what happens here, this is the process where NAD and FAD, they picked up hydrogens, they get rid of their hydrogens, and they pump them into what's called the intramembrane space of the mitochondria. We're going to label this another day, so don't worry so much about where that's located. The hydrogen ions will flow through an enzyme called ATP synthase. If you did the lab, which everybody should have done by today, you know that ATP is actually made in both photosynthesis and cell respiration by an enzyme called ATP synthase. That enzyme will generate ATP, and at the very end of the electron transport chain, oxygen picks up those hydrogens and forms water. So you are required to have oxygen or some kind of an electron acceptor like oxygen to run this type of respiration.